Well, thank you all for joining us today, both online and, of course, here at the Herschel Smith building. For our presentation today, it's my great pleasure and privilege to introduce Duncan Astle, Professor of Neuroinformatics here in the Department of Psychiatry. Duncan's only been a member of the department for a short while. I think you joined last year, is that right? September last year, yeah. But many people will know him from the MRC CBU, where he worked for many years. I'm not quite sure how many, quite a few. Wowzer, okay. And where he remains a group leader, yeah, so obviously made a big impact there. So broadly, Duncan's work is about brain development, I think I can say that. And one of the mysteries of biology is why we're all so cognitively different, including identical twins. So it's not all in the genes. So Duncan addresses this difficult problem with a range of innovative informatics approaches, some of which we're about to hear about. Duncan.
Thanks very much. Thank you all for being here online or in person. As John said, our research really sits somewhere between child development and neurodevelopmental conditions on the one hand and in informatics in the other. And I'm going to hopefully try and give you a broadish overview of the sort of work that we do. So as I'm sure that lots of people in the room already know, neurodevelopmental and child psychiatric conditions are really pretty common. I've said 15% there. Who knows if that's right? Depends entirely where you live, depends how you define it, but I think somewhere around 10 to 15% is probably a reasonable estimate. They involve a wide range of impactful cognitive and behavioral symptoms, often with lifelong consequences, and not exclusively, but mostly the cause is unknown. And in our group, we're particularly interested in the role that the brain might play in the onset and maintenance of those difficulties. And we use a variety of different techniques, sometimes called neuroinformatics, but they provide us with a kind of empirical framework that allows us to work across the different levels of analysis, all of which might be relevant in some way for understanding the onset and maintenance of those kinds of difficulties. And in the next 45 minutes or so, I'm going to try and persuade you that we can use this sort of framework to address some key challenges in understanding the developing brain. So the first one is about how we map heterogeneity. The second about how we build models to try and capture developmental processes. And the third one is about how we bridge scales and species. And the methods might be a bit unfamiliar to you, but the questions at the heart of it are going to be hopefully very familiar. So how do we map differences between children? Can we build models that explain those differences? What fundamental mechanisms might drive those differences? So even though the methods might be unfamiliar, the questions at the heart of it are some of the oldest, I think, in developmental science. And to introduce the problem and challenge and opportunity of heterogeneity, I want to introduce you first to three of the participants from a study that I'm about to tell you about. And this is where we find out whether the video works. I might just turn the volume down ever so slightly, otherwise we'll be deafened.
I just sort of really hard to concentrate.
English is like harder because in writing and reading, my reading's like not very well.
No,
Okay, so Freddie, Billy, and Eva, let me tell you a tiny bit about them. So Freddie experiences difficulties with paying attention. He's described as fidgety, always on the go. He described himself, when we met him, as disruptive at times. He has difficulties in maths, had lots of support in maths, and he has a diagnosis of ADHD. Billy also has real difficulties paying attention. He's received support across multiple subjects. He finds it very easy to be distracted. He experiences very high levels of anxiety. He has a dog called Buster. He has no diagnosis, or at least he didn't when we saw him. Eva has difficulties in reading. She's received lots of support in speech and language. The woman you could hear in the background there is her mum, who's also a speech and language therapist. She also experiences very, very high levels of anxiety, terrified of loud noises. She has a diagnosis of dyslexia. And we're interested in a broad set of questions about kids like this. So one cause or many causes. So Freddie and Billy both experience inattention, right? Is that same underlying cause of both their inattentions? Or is it different, slightly different causal mechanisms giving rise to a surface level set of symptoms? Which difficulties are most likely to co-occur? So for example, Eva's profile of struggling with reading, speech, language, and anxiety. do those things co-occur more often than you would have expected by chance? Or is it really just a chance occurrence in the case of Eva? And is the diagnosis relevant? So Freddie has a diagnosis, Billy doesn't have a diagnosis. If you were a teacher in a classroom and those two kids came into your class, does knowing that one of them has a diagnosis and the other one doesn't mean anything in terms of what you should expect. But it can be quite hard to address these questions, even though they might sound quite foundational. And that's because the kind of framework that we, and I say we in our lab as well as many others, typically use. And it's a paradigm that we're all very familiar with, I'm sure, the case control design, right? So the idea is that you choose a group of interest, you group the individuals with that particular diagnosis, and then you form a control group somehow, compare the two groups on whatever measure of interest it is. And whatever the difference is, you assume or you infer that that is somehow the underlying driver of the differences between the two groups. Problem with this approach, which I'm sure is familiar to many of you, most kids don't have a diagnosis, right? So for instance, someone like Billy would never get recruited to a study like that. So the majority of kids out there who are experiencing those cognitive and behavioral difficulties would never find their way into a study because for whatever reason, they haven't received a diagnosis. We often have to exclude most kids with co-occurring needs or diagnoses. What you're aiming for is the kind of pure sample. But of course, the reality is that co-occurring needs and diagnoses are extremely common in neurodevelopmental conditions. In fact, they're more often the rule than the exception. So excluding an awful lot of kids. And of course, it ignores the variability within and the overlap between the diagnoses. So for example, Freddie and Billy might have quite similar characteristics, but might find themselves in different diagroups. Two kids with ADHD might differ radically from each other. And this idea of grouping the individuals in this way and viewing neurodevelopmental differences purely through this lens gives us quite a distorted view about the underlying reality of difficulties that occur out there in the real world. And so for that reason, in the last 5 or 10 years or so, there's been a real push within the literature to try and complement this kind of approach, which still has its place, but complement it with a sort of transdiagnostic approach, where we set to one side the formal diagnostic labels that kids have, and instead try and focus on the impactful characteristics that sit just beneath. So one example of that is a study called CALM, which many people in the room will be familiar with. CALM at its heart is a cohort of about 800 children who are described here as being at neurodevelopmental risk. And that's because they've been identified by multiple different referral routes, child and adolescent mental health services, pediatrics, ed psychs, clin psychs, specialist teachers, occupational therapy. Basically, if that specialist comes into contact with children that they think could be experiencing cognitive or behavioral difficulties, they can refer the child to the study. They don't have to have a diagnosis, but they can. In fact, they could have multiple diagnoses. So it's a kind of all-comers study, intentionally broad. So yeah, they come with single, multiple, or no formal label. They're all in mainstream education. They're mostly age 7 to 11. 2/3 of them are boys. If there's any epidemiologists in the audience, you will instantly recognize that profile. So even though we're not recruiting by a diagnosis, it's interesting that that's still the kind of the group that you tend to see most. Okay, so when they come to see us, they're only too delighted to spit in a pot. They get invited for multiple different types of brain scan. They get around 4 hours of cognitive assessments, multiple different types of cognitive assessment, about 4 hours. Whilst the parents are captured, they do lots of behavioral questionnaires, measures of learning, measures of mental health. And as you can imagine, that creates a really, really rich data set. And so there's already about 75 projects. We've just finished the 5-year follow-up with about half the cohort. And any day it will be a managed access database so that you can go online, give a little bit of information about what you're interested in and what type of data you would want. And hopefully, touch wood, it should come to you relatively quickly. Okay. So there's loads of ways that you can use something like this, a data like this, to explore this issue of heterogeneity. And I'm just going to give you one example. And it's using a type of unsupervised machine learning called a simple artificial neural network, which sounds fancy, but is, as you will see in a moment, really very simple. And I will illustrate how it works with these fruits. So let's forget kids for a second. We've just got lemons, melons, oranges, and so on. And let's imagine that we could parameterize, we could assign values to each piece of fruit. We could describe it with 3 values, and perhaps size, acidity, seeds. We start off with a really simple network. And each node of our network, each circle, has 3 values associated with it. We call them weights in this context. So weight 1, weight 2, weight 3. And those, even though they're called weights, they're exactly the same things as the values. And so we initialize the weights, we give them some initial plausible values, and then we start the learning process. And the way that it works is that we take the first item of fruit and we assign it, we find the node in the network that has the weights that most closely mirror the values for that piece of fruit. So we have 3 weights, 3 nodes, each one described by 3 weights. And so essentially what we do is we find the node in the network that the first item is the closest match for. So where its values most closely aligned with the weights, so let's say it's here for argument's sake, we allocate the piece of fruit to that node. And then with a simple sort of error-driven learning, we update the weights. So they become a little bit more like those for the lemon that just went in. We take the next piece of fruit, find its best matching unit or node, allocate it, update the weights, and so on. Next piece of fruit, in it goes, update the weights. And so gradually over time, what we're doing is we're adjusting these weights to try and optimally represent the fruit data set. And what you will expect to find is that after some initial learning, you might find something like this. So it's learned that the watery things all sort of group together. There's a sort of category in there somehow of some kind of watery things with lots of seeds. It learns to kind of group these things together, these small sweet things, and you can see it's got a node for those kind of citrusy things. So what it's trying to do is optimally adjust the weight so that it kind of captures the variability in the sort of multi-dimensional fruit data set. The neat thing about this network is that it can adjust its structure as it learns. So you can see here that it can't distinguish the citrus fruits from the apples and pears, and so on the next time around, it might spawn an extra node. This time, it learns to distinguish the citrus fruits from the apples, pears, guavas. So that's the basic idea. And another neat little feature of the network is that it can spawn its own sub-layers. So at some point, it might realize, for example, there's a really noisy category in here, and to optimally represent it with the granularity you've requested, I'm going to have to spawn an extra little sub-layer to capture it. And you might think, well, that's jolly interesting, but why would you bother using an approach like this? Well, the first thing is that you might learn something. So the whole idea of an unsupervised algorithm like this is that it's trying to learn something about the underlying structure of the data itself. So we've got this really interesting, weird sort of multi-dimensional data set. In Calm, we recruited these kids in a kind of unconventional way. And the question is, who is in the sample? And the whole idea of this approach is that it tells you something about the underlying nature of the sample. And the second thing is it can create a model. And as we'll see in a minute, that can be a really useful way of making predictions. So we have a network like this. Obviously, we're not using fruit. We've got kids. And we're using the information from the cognitive assessments, so measures of memory, vocabulary, listening skills, attention, and so on. And the idea is that we're going to train the network up to try and represent those cognitive profiles. And the question is, what will the network learn? Maybe it will learn a kind of structure that we expect, or maybe it will learn something unexpected. Okay. So there's many ways of me showing you what the network has learned, and this is just the way that I find intuitive. And so what I've done here is show you the network from above. So you're looking down on it. And I'm showing you the network plotted in Euclidean distance space. That simply means that 2 nodes that are closer is because they have very similar weights. They're representing very similar children. 2 nodes that are a long way away from each other, they have very different weights. They're representing very different children. So we're kind of mapping out the space that captures the sample. And the network first did this kind of top layer, just 6 nodes in dark blue. So it kind of mapped out the coarse space. And then the yellow layer went beneath, and then the red layer underneath that. And we can now explore the weights of these nodes to tell us something about the kids that went into the network for the training process. So what we learn, for example, here we go. What we learn, for example, is that actually, even though the kids are referred for struggling, there are a group of kids in there that when you get them one-to-one, they have actually really good cognitive performance. And as you move left to right, you find pockets of kids with entirely age-appropriate cognitive skills, slightly better or worse at spatial or verbal things, but basically about a quarter of the Calm cohort are entirely where you would expect them to be for their age or better. Which is an interesting observation in itself. And as you move left to right, you find things really fan out. So the variability increases. You find a pocket of kids up here who really struggle with things like matrix reasoning, spatial short-term memory, spatial working memory, and so on. I've said spatial cognition difficulties here, but an alternative would be something like executive function difficulties, and it would be a totally plausible label for them. Down here, you find another group of kids. They really struggle with things like phonological awareness, like anything to do with rhymes, for example. They struggle with forwards digit span, backwards digit span. Their parents also describe them as having difficulty with syntax. So it's kind of like a messy language difficulties group. And then round at the bottom, you find a group of kids who basically struggle with every assessment that we give them. They're about 2 standard deviations below where you would expect them to be for their age. So what we've done is sort of trained the nodes up, and they've kind of spawned out to try and optimally represent the space. So an obvious question next is, well, what predicts your location in that space? And one option could be something like your diagnosis. So let's say, for argument's sake, that having a diagnosis of ADHD means that you have executive function difficulties, right? That's one of the dominant theories of ADHD. If that's the case, then we would expect those kids to disproportionately sit somewhere up here in the network. After all, that's where the network is representing the cognitive profiles. However, if having a diagnosis of ADHD in this kind of data set tells you nothing about your cognitive profile, then you'd expect those kids to be randomly scattered throughout the network. And of course, that's exactly what you find. And that's actually what you find for most of the kind of diagnoses that we have within CALM with large numbers. And that leads to a kind of simple conclusion, which is that in this kind of mixed data set, diagnosis itself is not a good predictor of a child's cognitive profile. In fact, we've done a lot of stuff like this, looking at different behavioral characteristics and all sorts of things. And ultimately, in a mixed data set like this, the diagnostic label per se is not a great predictor of child's behavioral or cognitive profiles, which we can discuss why that might be later. So, well, what does it predict? Well, one thing we can do is use some of the data that we've held out. For example, all the learning measures, the literacy, numeracy, spelling measures. And we do something called cross-validation, which I won't bore you with, but the basic idea is we train the network up with 80% of the network of the kids, and then we see if we can use that network to make a prediction about kids who have been left out, the 20%. And so we could see, for example, if we knew where kids should sit in the network in the cognitive space, how good is the prediction it makes about their learning profiles, relative to if we just plucked people at random. And when you do that, you find that your Cohen's d, your prediction using the space, is better than it would be by chance with a Cohen's d of about 0.5, right? So you make a decent prediction, a decent stab at what their learning profile should be. If that didn't work, I think we would really worry that something had gone seriously wrong, right? So next question, what about their brains, right? So we've got multiple different types of brain scan. One type of brain scan that we have is a T1 weighted image, which looks at the kind of outer 6 mil of the brain cortex. And we can explore whether differences in the gray matter in these regions that I've highlighted here aligns well with a child's location in the cognitive space. Again, the same holdout cross-validation idea. If you're interested in why we chose those brain regions, then ask me about it at the end, but I'm not going to go through feature selection now because it's really tedious. Anyway, so when you do that prediction, your prediction drops to a Cohen's d of about 0.15. Right? That's still significant because we've got hundreds of children in the analysis. But the point is that it's really small, right? Whether a child has a bit more or less gray matter here or there doesn't tell you very much about their cognitive profile, at least in this data set. Okay, another type of brain scan we have is a diffusion-weighted imaging. You guys, I'm sure, will know, but the basic idea of a diffusion scan is that we track the diffusion of water molecules. And you can imagine that if there was an invisible pipe, the water would diffuse nicely along it rather than through its walls, and thus I could infer the presence of the pipe, even if it's invisible. And that's the basic principle for the imaging technology, right? So we can infer the presence of these tracts, and we can use them to build something like this, which is called a connectome, which is a sort of simple wiring diagram of the brain at an individual level. And anyone who's studied connectomes in human beings will know that they have a quite characteristic organization. They're not random, and it's something we sometimes call small world. And that's because you get clusters You get clusters of nodes that are highly connected, and you get these hubs so that you can easily get from one part of the network to another. And we can capture that organization in multiple mathematical measures, capturing things like integration, modularity, centrality. And to cut the story a bit short, we could then explore whether these characteristics align well with where kids sit in the cognitive space. And the answer is that the role of certain key nodes, sometimes called hub nodes, seems to be particularly important. It makes a really good prediction. And that's because it's doing something pretty crude and simple, which is simply that kids who have slightly more small world networks are more likely to have better cognitive performance. They'll be more towards the left of the map, and kids who have more random networks will be more towards the right of the map. And that's a quite robust individual differences measure, and so it works well. Okay, so in the first section, what I've hopefully shown you is that we can use these sorts of techniques to build maps of the variability and then build models of the brain to test what might predict that heterogeneity. And it, of course, begs the next question, which is, but where does that variability come from in the first place? Okay, and for explaining that, I'm going to move on to the second part. Okay, which is using something called generative network modeling, which I will attempt to explain now. So let's imagine for a minute that I scanned everyone's brains in the room, and I built a connectome for everyone. Could I simulate how that connectome might have formed over time by adding connections iteratively, step by step? And because I'm setting the kind of mathematical conditions of the simulation, I might learn something really fundamental about what the constraints are on how a human brain organizes itself over time. And if I were to tweak those mathematical conditions ever so slightly for each person, could I optimize my simulation to capture their brain? And might the way that I have to tweak it tell me something really fundamental about the constraints that operate on all of our brains and might drive some of the differences we observe? And that's the logic behind generative network modeling. And at the heart of it is a really simple wiring equation, which I'll explain. So let's imagine for a minute we have two brain areas, brain area I and brain area J. And over time, we could express the probability of them connecting and wiring together as a trade-off between two things. The first thing is the cost. So if I and J are a long way away from each other anatomically, that's going to be a really expensive connection to form and maintain. If they're really close, it's going to be cheap to form and maintain. And I can trade that off against what I've called here value. Value is any non-geometric property of I and J. So for example, let's imagine I and J are a long way away from each other, but they all have multiple neighbors in common. They're all connected to similar nodes. If that's how I've defined value, shared neighborhoods, then even though there's a big distance, in this particular equation, those two nodes might wire together. And I've got these two modifiers, eta and gamma, and they just allow me to tune the contribution of the cost part and the value part of the equation. And if you're interested in learning more about that, then these three are the kind of foundational papers that I've put here. So this first one, the Kaiser and Hill project, they basically focused on the role of cost penalization or minimization in how brains form. Petra and Ed, they introduced the equation that I just described to you, this trade-off idea, and then Rick Betzel's paper here focused on comparing different value terms and tuning the model differently. So if you want to learn more, those are the ones to read. But you hopefully know what you need to know for this talk. Okay, so it's a bit esoteric, so here's a little worked example. Right, let's imagine we've got a little network, just 7 nodes. We want to simulate the formation of its connections. So early on, node 2 is about to form a connection. Right. And let's say, for argument's sake, that it just wires to something that's nearby. And it does that because there are no other connections in the network. So to start with, the wiring is just driven by the cost part of the equation. Right? There is no value to any node. Let's say it wires to node 36, which is really close. And you can see if I look at the distances between the nodes, then they perfectly mirror the wiring probabilities. The nearest thing is the thing that it wires. Okay. Later on in development, you can see that now there's lots of connections. There's some topological information, as we call it. Node 2 is about to form another connection. One option is it could form this cheap short-range connection with node 27. Alternatively, it could lay down this long-range connection to node 59. Now, because of the way that we've set up our rule, the trade-off, what it actually does is it forgoes the cheap short-range connection and lays down the long-range connection because node 59 already shares 2 neighbors with node 2, right? So because that's how we've defined value, it's deemed a high-value connection. And with this particular calibration of the trade-off, it forgoes the cheap short-range and instead lays down the expensive long-range connection. And you can see now, if we look at the distances, they don't neatly mirror the wiring probabilities. And it's because of this value term that's counteracting it. So you can see that these networks have this kind of nice property where early on, the network's primarily forming based on the cost of connections. But once there's some topological information in the network itself, that starts to drive the future wiring probabilities. And if we were just to look at that 7x7 matrix, the 7x7 wiring probability matrix, right? So you can see this is the connection that's really likely to form. And then when the connection goes in, if we form that matrix again, you can see now suddenly there are some very different connections that now suddenly become more likely. And that's, of course, because as soon as that connection goes in, the neighborhood structure of the network updates. New things suddenly find that they're neighbors. That adjusts the trade-off, and different connections become more likely. And so you can get this kind of stochastic process by which the networks can get steered down a different trajectory. That's the basic idea of generative network models. So this is work done by Dan Akaja, who was a PhD student, now a postdoc in the group. He used the equation that I've described to simulate the connectome for each child in the CALM cohort. So we're going to simulate the connectome for each child, and then we want to compare the simulation with the real thing. And we do that using an energy equation, which I'm not going to bore you with. But the basic idea is if it's a high energy simulation, that means it's not very good. That means the simulation is very different to the real. If it's a low energy simulation, that means that the simulation and the real network are very similar in their organization. So the idea is that you simulate the connectomes for each child, but remember, we've got these eta and gamma terms. So each time we simulate it, we're trying to tune it, we're tweaking those terms to see if we can improve the quality of the simulation. So we evaluate the energy for different combinations to see if we can find the sweet spot to make a really good simulation. I've given you one example for a K-term, this matching neighborhood idea, but there are lots of different terms we could cycle in there. Again, we put a new term in, we repeat the process, we compare the energy to the different K-terms to see whether by changing that term, we can get a much more realistic simulation. And when you do that, you find that some K-terms do much better than others. And if I can get to work, there we go. The very, very best ones are down here on the left. These ones in red, they're the lowest energy, and they are members of a class of model called homophily, which simply means that things will wire to things that are similar to themselves. And in fact, the very best one, this matching rule here, is the neighborhood matching rule that I gave you as the example, right? So when you have that as your K-term, you trade it off against distance, it produces really realistic connectomes. In fact, they're so realistic that if you had a batch of the fakes and a batch of the reals, you would really struggle to train an algorithm to tell the two things apart. Very plausible. Okay, but there's a bit of nuance, right? So remember, we were tuning it as we go. So it turns out that the really good simulations don't just come from any set of parameter combinations. They come from quite specific parameter combinations. So if you look at all the different gamma values we tried and all the different eta values that we tried, the low energy simulations, the ones in blue, all come from this tiny blue slither, this kind of cleft. They're all found in there. And if you zoom in for a minute, each one of these X's corresponds to one of the children in the CALM cohort. So essentially what it's saying is that tiny adjustments in the sort of economic trade-offs that governs the formation of the connectome are needed in order to capture the differences that exist across the children's brains. So implying that there's a slightly different set of economic conditions, the trade-off between costs and values is slightly different for each child. And of course, as I showed you, tiny amounts of variability in this trade-off can generate diverse trajectories as they cascade over time. Okay, so what factors might shape the network? So this has been a project by Alicia, a PhD student in the lab, and she's interested in the role that genetics might play in adjusting that trade-off. And so she used a data set called the ABCD cohort, which you might all be very familiar with. It's a US-based cohort. Neat thing about it is it's big, and they've got DNA data as well as neuroimaging. And so she used the DNA to create polygenic risk scores. I'm not going to go through the detail of how you make a polygenic risk score because I've never made one. But the basic idea is you're adding up common variants to try and explain a particular phenotype. And in this case, it's cognitive ability. And it explains something like 4 to 5% of the variance in cognitive ability, which doesn't sound like a lot, but in population terms, that's actually a large amount. Okay, and then she built structural connectomes for over 2,000 of those children. She fit the model, just like I've explained to you. And then you can imagine that for each child, we have an idea of the trade-off that governs their simulation. And then what she did is to try and establish which genes are associated with each part of the equation, the cost part and the values part. And she did that using postmortem data from the Allen Human Brain Atlas. I won't go through the details, but what you're basically doing is saying, for the distribution of costs, are there any genes who have an expression profile that is very similar to this distribution? Same for values. And that ultimately gives her 3 sets of gene lists. She's got the genes from her original polygenic score. She's got the genes that seem to be overlapping in their expression with the cost part of the equation, and the genes that seem to be overlapping with the values part of the equation. And what she can then ask is, are these 3 basically independent sets of genes? Are they sort of randomly scattered through the genome? Or is there some significant clumping? Do they group around particular cellular or biological processes? And she does that using a pathway enrichment analysis. And when you do that, you find that you do get some clumping, right? And so you can see that there are some functions like synaptic and peripheral membrane-bound cell development, and some stuff to do with transmitters and channels, where you get this kind of coalescing of the genes from the polygenic score in light blue, and the genes that seem to be associated with the values part of the equation. And so one possibility is that some of the variability that exists across all people in terms of the genes that we think are involved in cognition, also involved in the kind of trade-off that you see in the model. Okay, so that hopefully is the second part of the talk done. So we can build models to try and explain some of the variability in development or how it might arise. How long have I got, John? Okay, great. I might skip the very final bit, but I'll get through this bit. Okay, so next bit about bridging scales and species. So another thing that might be important for understanding brain development is the environment. Really, really hard to study the environment in children, having tried for many years. And that's because it's really hard to isolate actually what is the environment versus what is something else. And with that in mind, Sophia Carrozza was very kindly, by collaborators in Yale and NYU, sent a data set from 50 mice that they had collected. And a crucial ingredient, a crucial detail of this data set is that half the mice had been subjected to an early life stress paradigm, where they're removed from the mother in an unpredictable way, and the bedding materials are more scarce. And people who do this kind of work reliably inform me that that's a really robust way of creating a stressful environment in early life for the pups. Later on, they're all scanned. Sophia fits the model. She builds the connections first, which is not straightforward. She fits the model. Turns out the homophily models do really well. And if we zoom in on the energy landscape, you can see that tiny blue sliver, a bit like what we saw in the kids. And if we zoom in on that, what you can hopefully see is that the adversity mice are a bit more towards the bottom right, the control mice a bit more towards the upper left. This kind of continual horizontal axis, diagonal axis, sorry. And if you're more towards the 0 point, more towards the bottom right, you're more likely to be an adversity mouse. If you're more towards the top left, you're more likely to be a control mouse. And the difference that this means for the network is that when those parameters are weaker, when you're more towards the bottom right, it means that the wiring simulation is much more variable. At each step, it's less clear what the next connection is going to be. And if you plot the variability in connection here, you can see that quite clearly. The red mice are the adversity mice, and the yellows are the controls. And let's just explore that idea for a minute. So it seems to be that the successful simulations for the mice in the adversity condition have weaker wiring constraints on their preferences. And you can imagine if we had a little kind of toy network, if we had really strong constraints, you might have something like this, where as a step, it's really obvious what the next connection is going to be. If the constraints are weaker, then it will look more like this, where at each step, there are lots of different possible connections in play. And so what it's essentially saying is that the adversity mice are more towards down here. They're a little bit more like this. The control mice are more up here, a little bit more like that. And to see what that particular difference does to the way a network forms, what Sophia did was simulate multiple networks starting in kind of the optimal portion of space and gradually working down this diagonal until she gets down to 0. And when you do that, what you find ultimately is that your networks go from these small world networks that I've described before, and they gradually get a bit more random to the point that it's very hard to distinguish them from random networks. Okay, in short, adversity seems to weaken the constraints on network formation, and this makes networks form with more stochasticity and the resulting networks a bit more random. And you might imagine that if you're growing up in a much more unpredictable or chaotic environment, then there might be an adaptive advantage to having a kind of more stochastic developmental process. Okay, so that gives you a little idea about how we can use this kind of mathematical framework to go from our core interest in kids to start asking some causal questions in the mice. And that got us wondering about cellular mechanisms of development. And so this looks like a cartoon, but it's actually an image from an electron microscope of a neural network forming on what's called a multielectrode array. So a multielectrode array is a small sort of 2 centimeter square array, and you seed it with neurons, and then essentially you allow the network to form. These are rodent primary cortical neurons. You can see it in this really fancy stain, which was done by our collaborators, Manuel Schroeter over in Zurich. And you have to take my word for it, but the basic idea is that because these arrays have electrodes in the floor, as the network forms and the neurons spike or they fire, we can capture that activity and use it to form something like this, a kind of functional connectivity matrix on the basis of that spiking activity. If you're interested in how you do that, then do ask me at the end. But what you can then do, which what Dan did, was then simulate the formation of these networks. So not brains this time, but the formation of networks as they form in real time on the dish. Again, you find that the homophily models do really, really well. But something we wanted to test this time around is how well do these models do at capturing the kind of local characteristics of networks, not just the kind of global characteristics of networks that you measure with this energy equation. And so for a minute, if you imagine that if I were to take each, gosh, this is quite small for you. If I were to take each node of the network and across nodes correlate different properties of those nodes, you find that they have a particular set of relationships to each other. So for example, if I were to correlate, say, clustering and betweenness centrality, you find that those two things should be negatively related, right? If a node is part of a cluster, it's also less likely to be between clusters, right? So you get this particular kind of characteristic pattern of organization. And if you look across the different simulations, then you find that this matching model, one of the homophily models, you can hopefully see, even just with the eye, that that is really similar to that. Way more similar than any of these, which tells us that this homophily model isn't just capturing global characteristics of neural networks, it's also capturing something about the local properties of how those statistics are distributed, which is kind of exciting because it implies that these sorts of rules don't just govern the way that the networks between our ears form, they also work on a kind of micro scale. So you can see that only the homophily models do really well on the energy equation. So they do really well on the energy model equation over here, but they also do well on this sort of what Dan called topological fingerprint down here. So they kind of do uniquely well at capturing the global and the local characteristics. But of course, the neat thing with these is that we can explore how that emerges from 7 days to 10 days to 12 days to 14 days. If we look at the energy landscape, you can see that tiny blue slither, which should hopefully remind you of what we saw in the kids. Okay, So we're kind of working across scales, and you can imagine all sorts of cool experiments that you could then do. So for example, people have argued that it's really important to have GABAergic interneurons for the formations of these networks. Well, now we could grow some, but we could block those neurons using something called gabazine, which is a GABA-A antagonist. And when you do that, you can immediately see that you change the activity that you see in normal cultures to these sorts of flat profiles and rapid spikes. And when we fit the models, you find that it has no impact on the cost part of the equation, but it really pushes down the gamma, the values part of the equation. As we learned with the mice, when you weaken those parameters, you increase the stochasticity in the process. So you push this distribution rightwards. You can imagine you can ask all sorts of questions now about using pharmacology or rescuing networks in gene knockouts. And so what I've hopefully shown you is that we can use this sort of mathematical framework as a way from bridging our core interest in humans to work in animals, to work at the cellular level. And there is a final part on artificial models, but I think that I'm going to skip that because I think that we're about at time, right? But the basic thing is that you can train artificial networks to perform tasks and constrain them in the way that we think that the human brain, the new biological networks are constrained. And when you do that, you create networks, artificial networks that grow also according to the homophily rule. So we can recreate the scenario in an artificial network, which tells us a lot about what might be driving it. And that's really reminiscent, of course, of what we saw in the biological networks. Okay, so working across these different levels hopefully tells us something quite fundamental. In this case, that complex global phenomena developing brains can be an emergent property of the local constraints that operate in brains. Okay, so that hopefully gives you a bit of an overview of the kind of work that we do. So we try and use these sorts of tools to work with data from children and adolescents to try and map differences. We try and then build models to try and capture those differences. And then we collaborate with people doing cellular work or animal work to try and see if we can learn something more fundamental about underlying mechanisms. If you're interested, you could find out more from our lab webpage. Big thanks to funders, and a massive thanks to the people who do the real work. Sorry to those who I've not had a chance to go through their projects. There are many other projects I could have told you about. Big thanks to collaborators, especially Petra and Ed, who have helped us so much through with the modeling, and the CALM team who collected that really incredible data set for us to use. And I will stop there. Thank you all very much.